Digestion and absorption of carbohydrate and protein. Now, carbohydrates, you can look at them in terms of, first of all, sizes. So dietary carb sizes, there's three sizes. There's monosaccharides, which are the smallest. There's oligosaccharides slash disaccharides. And there's polysaccharides, such as starch. Now, the monosaccharides, because they're the smallest, they're easily absorbable by the small intestine. However, and some of the polysaccharides, you can digest two monomers to the monosaccharides and then absorb them, whereas other polysaccharides, they're non-digestible. You can also look at carbohydrate dietary fibers in terms of solubility, so they can either be insoluble or soluble. The ones that are insoluble, they're not fermented, and they include things like cellulose, which is a bulk laxative, as I talked about, which aids elimination in the gut. So even though you can't digest it, it aids elimination and maintains colonic health. On the other hand, soluble dietary fibers, they're easily digested by colonic bacteria, and they include pectins, gums, and mucilage. Now, the dietary fiber, 45% of it is starch, and dietary fibers come from things like fruit, veg, and cereals. So dietary carbs, as I said, they're 45% starch, but let's have a look at what else makes up the dietary carbohydrates and what they're made of. So first of all, looking at starch, well, starch is for storage, and it's found in plants. It's a store of carbohydrates, essentially. And what does starch contain? Well, starch is made up of two things. It's made up of amylose and amylopectin. Now, what amylose is it's straight chain, first of all, and secondly, it contains alpha-1,4 linkages. Whereas amylopectin, it's branched, and it contains alpha-1,4 as well as alpha-1,6 linkages. Now we can compare starch to glycogen, because glycogen is like the animal version of starch. It's also for carb storage, but it's in animals rather than plants. And glycogen is similar to amylopectin here. It's similar to amylopectin in that it has alpha-1,4 and alpha-1,6 linkages, and it's branched. However, it's different in that it's more branched. Monosaccharides make up 5 to 10% of our dietary intake of carbohydrates, and the monosaccharides are fructose, galactose, and glucose. The small bowel can only absorb these. Remember, the disaccharides and polysaccharides, they're converted to monosaccharides to be absorbed by the body. And disaccharides make up 30 to 40% of our carbohydrate fiber intake. And 3 of the disaccharides shown here: sucrose, lactose, and maltose. Maltose is made up of glucose and glucose. Lactose comes from milk. It's the sugar found in milk, and it's made up of glucose and galactose. And finally, sucrose. Sucrose is found in sugarcane and some fruit juices, and it's made up of glucose and fructose. So when you eat carbohydrates, you need to digest them, obviously, and the digestion of carbohydrates takes place in 2 steps, right? The first step is luminal digestion, and this occurs within the lumen of the bowel. And the next step is membrane digestion. So this is what happens when it reaches the brush border of the enterocytes, and there's brush border enzymes that they're membrane proteins that digest them further, and that's called membrane digestion. So there's luminal digestion and membrane digestion. So let's have a look at how that happens. So luminal digestion happens with amylases. So first of all, you eat the starch. Let's say starch is a polysaccharide. The first thing that happens is salivary acinar cells, they produce salivary amylase, right? And that initiates the digestion of the carb. But then what happens is that the salivary amylase is inactivated once the food reaches the stomach. It's inactivated by gastric acid. So then what happens when the food passes from the stomach into the small intestine? The pancreas secretes amylase, which completes the starch digestion in the small bowel lumen. Where does the pancreatic amylase come from? Well, it comes from pancreatic acinar cells. And what causes the pancreatic acinar cells to secrete amylase? Well, it's cholecystokinin. Cholecystokinin, acetylcholine, secretin, and VIP, but cholecystokinin is the main stimulant. Where does cholecystokinin come from? Well, you remember cholecystokinin comes from duodenal eye cells in response to fat, right? And this luminal digestion by the salivary amylase and the pancreatic amylase, essentially it breaks down the starch into maltose, maltriose, and alpha limit dextrins. So what these are, you'll notice, they're not monosaccharides, are they? They're not sucrose, glucose, and galactose. So therefore, you can't absorb them yet. Instead, they're oligosaccharides. So they're smaller than the polysaccharide, but they're still not absorbable. So maltose, maltriose, and alpha limit dextrins, these are oligosaccharides. And this whole thing by amylase, this was luminal digestion. So once you've had the luminal digestion into these oligosaccharides, you then need to break it down further into monosaccharides. The way that happens is that on the brush border, so that's the border of the enterocytes from the lumen to the intestinal cell, brush border disaccharidases. So ases implies it's an enzyme. Disaccharide / oligosaccharide implies that it's an enzyme that deals with oligosaccharides and disaccharides. So this is what it does. Well, the disaccaridases, they're membrane proteins, and they hydrolyze the oligosaccharides, maltose, maltriose, and alpha limit dextrins. And there's 3 disaccaridases on the brush border. First of all, there's a lactase. Now what lactase does is it breaks down lactose, which is a disaccharide, into glucose and galactose. Maltase, another brush border enzyme, it deals with alpha-1,4 links, right? So what it does is it converts maltose to glucose and glucose. The next enzyme is called sucrose isomaltase, and it's made up of 2 enzymes essentially. So the sucrase part of it that converts sucrose into glucose and fructose, and the isomaltase part of it that's important. The reason that's important is to deal with these things, these alpha limit dextrins, which is an oligosaccharide. So what it does is it deals with the alpha-1,6 linkages of the alpha limit dextrins and degrades them. And this whole process, because these are on the membrane, this is called membrane digestion. And 2 things to note here, I guess, is that lactase, which breaks down lactose into glucose and galactose on the brush border membrane, you can have a lactase deficiency. So if you have a lactase deficiency, obviously this breakdown is not going to happen, first of all. And secondly, the peak activity of these brush border disaccaridases is in the proximal jejunum, so that's immediately after the duodenum. And you can see that here. So where are the enzymes active? Well, the brush border disaccharidases, their peak activity is in the jejunum. It's less in the duodenum and ileum, which is either side of the jejunum, and there's no activity in the colon whatsoever.